In the previous video, I said that bioinformatics is in fact a combination of 4 different disciplines, and that's why it's so challenging: math, statistics, biology, and computer science. Today I want to emphasize a little bit on the importance of statistics in bioinformatics, and I want to say that statistics is so important for bioinformatics that in fact bioinformatics is nothing else than just applied statistics on biological data. And in fact this sentence here contains everything else, because we need math in order to develop the models, to use them in statistics, and we also need biology to provide the data, and we need computer science to develop the software that statistical methodologies will be applied on biological data. Okay, but in order to understand the importance of statistics on biology, we need to emphasize a little bit on what kind of data we have in biology in order to associate them with concepts of statistics. And the most important concept in statistics is the one of the random variable. So if statistics is so important in biology, we should be able to see biological data as random variables. Now I will bring you a simple example, a real world example, and then later on we will go to also genetics and see how genetical data can be seen as random variables. So imagine that you have a well-defined population here of students, let's say, and then we have student A, student B, student C, student D, and student E, and student F. Now this is our whole population, and from this population we can take a random sample, a randomly picked individual according to some probability rule. And let's say that for this population of students, what we measure is how well a student performed in a given exam. So this is a mark, an exam, and it's a discrete value between let's say 0 and 20. Now if we randomly take this A student here and we see his grade, then we might see that he has the grade 11. And then we randomly pick another student according to some probability rule, this one, and this student has the grade, the mark 14 out of 20. So generally speaking, the mark of a student can be, the specific mark that a student has can be symbolized with a letter, small letter M. So M here defines the exact value that a specific student has. But generally speaking, in this population of students, we assign a capital letter variable m that defines the concept of a mark. And the important thing for us to understand what a random variable is that for this mark here, for this concept of a mark, we cannot know the exact value of this m unless we pick a student and we see how he performed the exams. So, in other words, a mark is like a box, like a function. So the concept of mark is like a function that gets input as input a student and outputs a specific value m, which is the mark of the student. And we cannot know the value m unless we pick the student and see how well he performs. So this inability that we have to know exactly the value of capital M makes capital M being a random variable. Takes values. Okay. Now the nice thing with random variables is that they can combine between themselves. So if we assume that we have another random variable, let me use this color here, and this is the h random variable, which defines the hours that a student spent on studying. Then, for example, this student he spent 3 hours, so this in hours, and this student here spent 4 hours. And again, this h here is a concept of this specific population of student. Then again, we can imagine that h is a machine or a box that gets as input a student and outputs some specific value h, and its value we cannot know it unless we see which student we are talking about. We see the student and we ask him, okay, how much time you studied for these exams, and he tells us 3 hours or 4 hours. And again, it's a random variable because we cannot know a priori its value. We have to pick the student and ask him, and then we will know the value of h. Unless it's a very easy class that nobody needs to study, so for all students this value will be 0, and then we don't need actually to ask anyone how much time he studied, the answer will be 0. And then the h would not be a random variable, because we don't need to see the student and ask him in order to determine the value of h. But in this case, in the general case, we need to know the student in order to know the value. And the nice thing with random variables is, of course, they can combine and they make new random variables. So if we want to combine them, we can have a different axis here that we will call E from efficiency, and I don't know what units it will have, but we can call efficiency as the M, so the mark divided by H. This will be a new random variable called E, that again its value cannot be determined unless we know the student. So how we do that? If we pick student A here, then this will have value 11 for the grade, value 3 for the hours, so he will have the value 11 third as efficiency. And the other student here, student C, will have the value, I don't know which one is bigger, but the value 14 for as an efficiency, whatever it means. The important thing here is that we can create new random variables, and again the value of this new random variable cannot be determined before we know for which student we are talking about. So all these are examples of random variables that we can find in everyday life. And in order to study and make conclusions from these random variables, as we will see throughout the class, we use the tools of statistics. Now the important thing for us is that the biological data that we will use are also, we can see them as random variables. And I will just give you a brief example, and later on we will see that in more detail. So again, imagine that we have a population of individuals and what we study is the expression level of a specific gene X. So let's use the notation E here, capital E, to assign it to the concept of expression level for this specific gene. And now you can understand that the expression level E is in fact a random variable, because you cannot know the exact value of the expression level of the gene X unless I tell you which is a sampled person and measure its expression level for gene X. So again, we will have this X here, we'll have E that measures the expression level, and if we pick up this guy, we'll have the expression level 115.5, but if we pick this guy, we'll have the expression level 120. Now it's interesting that I think that this example is clear that the expression level of a gene is a random variable, but it's interesting to notice that we can assign more complicated biological data to random variables. So imagine that we sequence that now we don't talk about the expression level, but we talk about the genotype, capital G, of individuals. And I will claim here that also this is a random variable, because if we sample this individual, now I don't need to use an axe here, because this is not actually a numerical value. So if I sample this individual, then this individual might have the genotype ACCGTACGA, tiny genome. Imagine that this sequence for humans is about 3 billion letters, but this individual might have the sequence, the genotype ACCCTCCGA. So compared to this one, this genotype has a difference here and a difference here. These two genotypes are not exactly identical. So again, the genotype itself, even though it's not a number, it's a random variable that we cannot know it beforehand. We cannot know it a priori. We can only know it if we sample the individual and we sequence its genome. So again, the genotype is like a box that inputs a person and outputs the genotype, the value G here. We can imagine several random variables for the biological data that we will study throughout the course. So for this introductory lecture, it's important to understand that all the data that we have in biology, or more or less most of the data that we have in biology, are in fact random variables, and therefore we can apply statistics to study them. Thank you very much.